In this video, we will be examining the mechanisms of transcription in a prokaryotic organism, which is split into three simultaneous stages: initiation, elongation, and termination. The initiation stage is characterized by the binding of RNA polymerase to the promoter region, which is a specific DNA sequence that the enzyme binds to in order to initiate transcription. The promoter region provides varying transcriptional directions, but two promoter consensus sequences are conserved in prokaryotes, the -35 region and the -10 box. The -35 region contains a binding site of the sigma factor, and the -10 location, the TATA box. RNA polymerase requires the addition of a sigma factor to the core enzyme, producing the holoenzyme so that it is able to recognize and begin transcription at the correct initiation site. The subunit is removed once elongation begins. A general model of elongation reminds me of a worm on a string, in which the worm represents the mRNA with an elongated tail. I will demonstrate the specifics on the whiteboard. During elongation, the core enzyme RNA polymerase moves in a 3' to 5' direction along the template strand, starting at the +1 location, synthesizing mRNA in the 5 to 3' direction. The template strand of DNA provides a template of sequences for RNA polymerase to create mRNA pairs for, while non-template strand DNA is commonly called the coding strand and is complementary to the template strand. After transcription, RNA polymerase must dissociate from the template strand in order to release the mRNA by either row-independent or dependent termination. In row-independent termination, a termination hairpin forms as a result of inverted repeats in the sequence that bind together and cause the mRNA to fold in on itself. This folding phenomenon causes the RNA polymerase to dissociate, and the mRNA is freed. In row-dependent termination, the row enzyme binds on the 5' end and moves towards the 3' end of the mRNA. As the RNA polymerase pauses at the termination site, row collides with it, releasing the mRNA via helicase activity of row. Now, what if there is an insertion, specifically a 5 base pair insertion, in the promoter region between the -35 region and the -10 box? We have already discussed the mechanisms that are essential in the initiation process, and now we will hypothesize how this insertion will affect gene production. A hypothesis of this phenomenon is that an insertion would cause major shifts in a promoter consensus sequence locations, inhibiting RNA polymerase from binding properly. Without proper binding, there will most likely be less phenylalanine in the affected individual. Phenylalanine is especially useful in protein synthesis as well as dopamine production. An inadequate amount of phenylalanine may lead to a protein deficiency, depression, and other adverse health effects.